Hello and welcome to MMI. In this series of tutorial videos, we'd like to show you the basics of the MMI CellCut and how to get good results for further research. The MMI CellCut is our laser microdissection system designed to cut tissues, large regions of interest or single cells, and even living cells for further processing. With its high-pulse UV laser for precise cutting and the patented CapLift technology for reliable tissue and cell isolation, it will help you with all your laser microdissection needs. In this video, we'll show you how to dissect samples from tissue and also how to collect them with the CapLift. You will learn about the multiple collection methods and about the predefined target positioning, or short PTP. The first thing we need to do is to mark the structures we want to cut. We have to draw a shape. Therefore, we have multiple tools. One of them is the freehand tool. It has an option to automatically close the shape. Just draw your shape and it will be automatically closed if you check the checkbox. You can also draw circles. Select the tool, click and draw, and you will create circular shapes. You can also set a fixed diameter. Now a circle will be created around the point you click with the diameter you defined. The same will work for the tool for ellipses, where you can define fixed geometry. You can also draw straight lines with fixed length or rectangles. The last tool is the erasing tool. Select it, click on the shapes, and they will be automatically erased. You have also multiple deleting options by using the right-click menu. Just right-click and you can delete highlighted shapes, clear all groups, clear cuttable groups, and clear the current group. How to define group will be shown in another video. There's also the option to clone shapes if you need the same shape multiple times, for example. Herefore, you need to draw a shape and see that it's highlighted. Highlighted shapes are always brighter than the other shapes. Then right-click and click on Clone. Here you can select the grid size. You can define how many columns and rows of your shapes shall be drawn. And you can also define the spacing between the different shapes. Click OK and you have cloned your shape. They will form a grid. Now it's time for cutting. How to calibrate the laser was shown in a previous video. The settings are good, we have drawn a shape, and now we can click on Cut. Sometimes you might want to cut thicker tissue, and it could be useful to cut several times, then increasing the laser power. This can be done by increasing the repeat value. Then the shape will be cut several times depending on the repeat value. And you can also set a set drill value that will change the focus of the laser with every repeat by the defined value. But our cutout looks quite good, so now it's time to pick it up. In this case we use the manual mode. Just click on Collect and the CapLift will move down and up, collecting our cutout. Besides manually collecting your cutouts, you can also choose between two automatical collection methods. One is dissection with cap down, one is dissection with cap up, and both can be combined with clustered collection or one-by-one collection. In the clustered mode, the CellCut will first cut all your selected shapes and then collect them all together. In this example, we want to use the cap down method, which means that the cap is down during the cutting, so your samples can't get lost. We move the cap down to be in the right focus plane, and then we draw some shapes. Then we click on Cut. The CellCut will start cutting and will cut the shapes first and then collect them. In the one-by-one mode, the CellCut will collect every shape after it is cut, one by one. In this case, we use the cap up method. Then we click on Cut, and you see that the first shape is cut, then collected, and then the second shape is cut and collected. The great benefit of the automatic collection methods is the predefined target positioning, short PTP. This algorithm ensures that every cutout will be collected with another part of the cap by moving it in a spiral way, and avoids stacking of multiple cutouts on the same place and therefore losing some of your cutouts. You can also select different cap sizes with the drop-down field. Don't forget to set the system to select new caps after each dissection. There's one small thing to keep in mind when working with the CapLift. The cap needs to touch the membrane fully. If you are at the border of your slides, the software will show you a little warning symbol. This means that the cap cannot surely touch the membrane because it's too far on the border and will touch the metal frame instead. Sometimes you might want to cut shapes that are bigger than the view frame in the software. We do have a solution for that. Let's cut this bigger structure. We click on the freehand tool and check the checkbox for Extend Selected Shape. Now we can draw a multi-segment drawing. We start with drawing the first half of our desired shape. Then we can move the screen and draw the second half of our shape. We hope this video helped you with working with the MMI CellCut. Feel free to visit our other videos for more information about MMI instruments and handling.